chaperone.
Okay, so
Oh, you really meant chaperone.
chaperone. Yeah. I didn't know that it was chaperone me. For me, like, so you say proteome me too?
No.
No.
He's joking.
It sounds like salami.
When you're speaking like that, yeah. Okay, so I want to talk about the chaperone, but there's no chaperone here. That's the problem. So that's why I will talk about the chaperone project. And actually, it's Richard Morimoto, who is a professor at Northwestern University, who wants to do that. And so it was my teacher last semester, so that's why I know about that and I want to present this work. So what do we want to know? So we want to know all about the chaperones, but through a network approach like you have, like people have done for the proteome or anything. So just a few slides to recall what is chaperone and everything. So basically, chaperone is just a protein which helps other proteins to get folded correctly. That's really important because if you knock down one chaperone on E. coli named DRO-EL, E. coli cannot survive at all. So at least one really important protein in this cell has to be helped for the folding by the chaperone. So they have been first discovered as HSP, Hitchcock protein, to have the protein to get folded and not to be aggregated with other proteins. But also they have to let them some space, if I can say that, for the protein to get folded, because if it binds too much to the hydrophobic residue, they will not get into the hydrophobic core of the protein. So that's for most of them, that's why they use ATP, and it's really ATP consuming. Like if you take HSP60, just the first ring, just the higher part, you have 7 oligomers, and each oligomer by an ATP, so it's 7 ATP per cycle, and sometimes a cycle doesn't manage to hold the protein. So, but this makes small HSP doesn't use ATP, but they have shown that this type of protein helps the protein to get folded in redirecting them to other chaperones. So that's my other part in this slide. They have a lot of collaboration between them, and just to see the collaboration between different chaperones, also that it's a really major pathway in the lighting, and we can find a lot of homologous chaperones on the lighting. So what do we want to know? We want to know all the chaperones, but I think what's really important is to see that we want to know which chaperone is active in which cell at one time. Why? This is really important. It's because the chaperone helps all the protein to get folded, and it's not the same issue for a developing cell or a cell with becoming a muscle cell. Like it will produce a lot of actin and cytoskeleton protein, so it will be really stressful for the cell and for the chaperone's network, and also for the aging, because of course everybody has heard about the aging diseases like Alzheimer's or Parkinson's, and it's a matter of aggregated proteins in the brain. And they have shown that the chaperone network it's not that high, and that's why the chaperone network cannot manage to clear all these proteins. And also on the cell side, because in these diseases you just find in some cells, so you have this idea that some cells are more sensitive than in the others. And also you they have been shown that it's part of the striatal cell response. So we want to know which chaperone is active and how can they interact together in all these conditions. So that's what this slide is about. So we want to know about all of these interactions. So chaperones are really busy proteins. So just a word about the regulative pathway. So we know that there's a cell level regulation. Of course, HS1, the most known transcription activator of HSP genes in eukaryotic cell, but there's also other cells. But what is really important from my point of view is the organism level regulation, because that's why it's really important to use a whole organism to study at each time which chaperone is active and everything. And so I just talk about the insulin-like signaling. They have shown that in C11 some cells have insulin-like ligand receptors, like some neurons and gut cells. And in this cell, the insulin, when insulin ligand binds to this receptor, it will affect the HSP gene expression. Because when you have insulin, it means that you have a lot of sugar in your environment. So you are, yes, I want to develop myself. I don't want to be stressed and everything. So the HSP expression level goes down. But we don't know. We do know that it happens in these two types of cells. But how about all the other cells in the organism in the same condition? Is it sufficient to have just two types of cell who is responding to this signal? So how can we get that? Just really fast. So they want to identify genes which co-express in the same condition to have an idea of a network interaction, and after they want to have a step or more classical biochemical and biophysical studies to confirm or infirm these interactions. So that's what I wanted to talk about, and I hope I have told you enough to convince you it's really important to know what is going on in every cell, in every time, in a lifetime already. Thank you.
That was perfect, exactly 10 minutes. Okay, questions?
So can you also use proteomics to look at this question? I mean, it looks like you are talking mainly about transcription of different chaperones.
Yeah, I think that they want to do that first. It's just a project, so they want to start by that, and I don't know after what they can get or not, because they are not very clear about which type of computing analysis they can get and how relevant their network interactions hypothesis can be. Yeah.
So was he in Paris last year?
Yeah, just for 1 month. Because our school partner.
All right.